So this model will show what occurs when muscle contractions happen so that the body can move. So the skeletal muscles are composed of like muscle fibers, and the muscle fibers usually contract or relax to cause the movement, such as lifting objects up or kicking or anything that causes your body to use these skeletal muscles. And the way that happens is that they receive signals from the nervous system telling the body that the contraction needs to occur. So the neuromuscular junction, which is shown right here between the motor neuron and this muscular cell, so then it's right there, is the site of where that signal kind of shifts from your nervous system into your muscular cell. And so the fibers, which are composed of myofibrils, actually have contraction mechanisms or devices in them called sarcomeres. The sarcomeres are shown right here. These sarcomeres are made up of actin and myosin. So usually the sarcomeres form this interlocking chain which works like this. So when it contracts, it pulls together, and when it relaxes, it expands apart. And this occurs similarly throughout in several layers going on and on and on. And that's what causes a striated look on like a muscle, because you have these interlocking groups just lining up like this, and so you have these lines going across, and they're usually in a very parallel structure. So like I said, they run kind of in a parallel way, and they interlock. So, and they work from both sides. So if this is just one cell, but usually you have on top, bottom, left, and right, so where it interlocks on the sarcomere. And each sarcomere consists of like a thick part and a thin part. The thick part is the myosin, the thin part is the actin. And that's another reason that it has that striated look, because you have these varying bands of thick, thin, thick, thin, thick, thin. And so the thick filaments, which are the myosin, actually are pulling on the actin to cause the contractions. The actin, which are on the Z line, are on both sides of the myosin, right there and right there, are kind of pulled in by the myosin. It will grip at the bonding site, and it will pull, and it will pull, and then it will relax. That's what causes the contractions and relaxations. And so the pulling that the myosin does, it is able to do that by like using a force and at the site called the crossing bridge, or the cross bridge, excuse me. So the cross bridge is kind of where the bond, or I wouldn't say bonding site, but where the myosin grips the actin and starts to pull and pull and pull until it's fully contracted. The contractions usually are due to the sarcomere shortening from both sides. So not only is it pulling from this way, but this myosin is also pulling from both directions. So it's also pulling in from the actin that would be on this side. And so the contractions usually begin when ATP is hydrolyzed into ADP. So it's going from triphosphate to diphosphate. And that's caused by the myosin extending and attaching to that binding site that we talked about, the cross bridge on actin, and and a thing called a power stroke occurs, which pulls the actin which was on the Z line towards the M line, which is what the myosin's on, and it pulls off a phosphate group. So that one phosphate group is pulled off during what's called the power stroke, and that's when the force is exerted by the myosin onto the actin to attract it and contract the muscle. And so the myosin is the myosin is using the ATP, converting it to ADP in the power stroke, and so that phosphate travels from here to the 2 or however many myosin strips there are going on, and then that goes into the actin so that the actin can move. And so that continues on until all of the ATP is used for that run or that power stroke. Then the muscle relaxes. So the muscle contractions are actually controlled by the actions of the calcium in the thin actin filaments. And so the calcium is stored in the higher locations right there. In our diagram, we stored it further to the top right here due to structural differences of our molecule. So the calcium is actually a regulatory protein that the actin uses, and it helps with troponin and tropomyosin. So when the muscle relaxes, as in this position, the cross bridge, where the binding site, which is known as the cross bridge, where is where like the actin and the calcium interact, and that's when and that only happens when there's higher levels of ATP present, so that the calcium is able to tell the actin to actually bind to the troponin. And originally, what troponin and tropomyosin do are, tropomyosin inhibits the binding. So when calcium interacts with the actin, and when it starts to pulling, the troponin is responsible for enabling the tropomyosin so that the binding can occur, because tropomyosin is responsible for stopping the binding or inhibiting the binding, and the only way that the muscle contraction can occur is if the actin and the myosin do bind. So once the troponin acts on the tropomyosin to take away that inhibitor, then the binding site occurs, and that's how myosin is able to exert its force. That's how it's able to start pulling on the actin. The calcium ions are stored in the sarcoplasmic reticulum, which is shown here, and it's released when they get signals from the nervous system that contact has been made in the neuromuscular junction. So the neurotransmitter molecules actually come from a neuron, and they bind to specific receptors, which are actually in the membrane of the muscle fiber, so they're up here. The electrical impulse, what happens to the electrical impulse is that it travels down the T-tube, as you may or may not have seen, but we have electrical impulses which are traveling down the T-tube, and it opens the calcium stores, which are once more stored in this region up there by the sarcoplasmic reticulum, and it just kind of it tells the calcium ions to go to where the myofibril is and start triggering a muscle contraction, so that the actin and the myosin start interacting, and the sliding occurs. The entire muscle fiber shortens in response to the three proteins and the actions of the actin and the myosin, as well as the storage and buildup of ATP. Only when there's enough ATP can this reaction occur. So in a summary, I guess, the striated muscle fibers which make up the muscle cells, the muscle cells which make up the striated muscle fibers are composed of myofibrils, which are actually like filaments that are in a parallel pattern of sarcomeres. The myofilaments, which have the thick filaments, which are the myosins, and the thin filaments, which are the actin, and then also have troponin and tropomyosin acting on the actin and myosin, are the entire structure. So the actin and the myosin are the proteins that will contract to help the which will actually, contract to help the body or the muscle itself contract with each repetitive movement. And they work together by causing the binding site to pull on the actin from both sides of the myosin so that the contraction can occur and cause the muscle movement. Troponin and tropomyosin are regulatory proteins that that regulate, the work between actin and myosin. Furthermore, the myosin and actin together make up the sarcomere. The sarcomere is on the Z line for the myosin and the Z line for the actin and the M line for the myosin. And the thin filaments, once more, are the actin, and the thick filaments, the myosin. So now on to rigor mortis. Rigor mortis is actually the cell death, and it's like a stiffening of your muscles that stops the actin and myosin from forming the cross bridge, so that power stroke cannot occur. So in a living muscle cell, as we just saw, the two are able to bind due to troponin and tropomyosin, and also due to calcium and the ATP. But once the body dies in post-mortem, the body is not able to stay relaxed without having, it's not able to have enough ATP for rigor mortis so that rigor mortis does not occur. So there's actually 4 steps in rigor mortis. So during the first one, it's called, I believe, the delay phase, which usually lasts between the 6 to 12 hours, and it's kind of a permanent situation where the bonding of the actin and myosin cross bridge is postponed or delayed for that period of time, because there's creatine phosphate, one of the phosphates from the ATP, and that's not able to, or the creatine phosphate, which is actually responsible for taking one of the phosphates from ATP, is not able to make it from ATP triphosphate to ATP diphosphate. So what's occurring in that situation is the body is not able to have these contractions occur and have the cycle going on as fast as it should. And that's because the energy is not being able to be collected or formed, and the bond is not being able to be broken. So this causes the muscles to become really slow, weak, and almost, frozen in a sense. And so during the next phase, which is the onset phase, the muscles kind of become stiff and they can't be extended, and eventually all other positions for the muscles to be in starts to lose, because they're really tense muscles, because the movement is not occurring, the bonding is not occurring, and the ATP is not breaking up as fast as it should. And it's just really important to note that the bonding site is not it's kind of fixed, it's not bonded, but it's in that contracted position. And so also the pH of the muscle cell starts to decline when rigor mortis occurs, because the metabolism, which is the speed at which this reaction occurs, is very, very low. So since like I said earlier, the bonds are already the bonding sites, the cross bridges are formed, but the bonding is not occurring. The muscles are already contracted as far as they can be, but the actual process of contraction and relaxation is not occurring at the speed that it should. So the muscle and the conversion of ATP into the energy needed for the actin to be released once it's used up, since you're not really breaking down the ATP into ADP, there's no final energy position. Like the use up of energy is not able to occur, because in truth you are using energy, but there's no energy that's been broken or dissolved at that point. So the pH is lower, the energy is not being broken down properly, and the cycling of the process is very slow. Also, the tension, and then the last phase, finally, the tension kind of releases. Like I said earlier, it's very tense because the muscles are already contracted, but there's no energy that's being used up at the same, well, there is energy that's being used up, but it's not as quickly or as smoothly as the cycle should be in. And so finally, in the last phase, which is called the resolution phase, the final degradation, I believe is the word, like the body just kind of not decomposes, but it finally loses its ability to hold this position of the bond site or the cross bridge being there. So finally, that kind of breaks down, and the body goes into this tight final position, and that's the end of rigor mortis.